Good evening, everybody. Dobro veče. Actually, this panel will be in English, so I will welcome you all in English. We're happy to have you here tonight. We're organizing this panel as a part of Cost Action XRNA Path. But it's kind of really nice that it's timely related to Nobel Prize that was just recently announced in the field of medicine and physiology. But I won't be talking more about this because I don't want to reveal anything that will be talked about in this panel. My role was here just to welcome you, and I will introduce you to Katarina Stekic, who will moderate this panel, and she will then introduce you to our guests this evening. So, Katarina, you have your own microphone, or you want this one?
I mean, or I can just speak loud, whatever works best. Good evening, everyone. Those of you who are here today, welcome to the final event of this evening. And those of you who just came, welcome to the first event of this evening for you. This panel will be structured mostly around how we perceive RNA technologies and how they actually are, and what's the future of RNA, how we perceive science in general, and what we have about trust in science regarding our data so far. So I will just briefly introduce our panelists. Professor Iris Jajé from the Faculty of Philosophy. She is also the head of the Reason for Health project, which is funded by the, what's it called, the Fund for Science? Yeah. Fund for Science, yes. And she will be presenting some of the data that we have from the project that she is working on. We also have Stephen Fletcher. He's a bioinformatics specialist from the University of Queensland, Australia, working on crop protection and biopesticides. We have Amy Buck, who is from the Center for Immunity, Infections and Evolution from University of Edinburgh. Maja Kosanovic from the Institute for the Application of Nuclear Energy in Serbia. And we have Rob Lackey, who's a marketing consultant, if I'm not mistaken, communication specialist, currently working for Greenlight Biosciences. Welcome, everyone. Thank you for joining me in this panel. And I would like to start this discussion with briefly just focusing on what we have in Serbia in particular about trust in science. Where are we currently regarding trust in science, what we know so far, and then we can move forward to RNA technologies in particular. Iris, would you like me to put the presentation up?
Hello, I'm very glad to be here. Thank you for inviting me. I hope this will be interesting for you, because I'm a psychologist and I basically I'm interested in how people perceive science and how they develop trust in science and whether they are reluctant to accept some hot topic, contested topic when it comes to science. So I just tried to put it together in a couple of slides, and I'll be here for a longer discussion later. So I'll start with some good news when it comes to Serbia. It's regarding scientific literacy. Within our lab, we had the opportunity to test a representative sample 2 years ago and ask them 12 standardized questions about very basic scientific knowledge. And as you can see, the average is 8 out of 12. So people correctly responded to 8 out of 12 questions, and the educational and age differences were as you might expect. So to zoom in the questions so you can have a feeling of what we asked them, it's very, so to speak, basic knowledge, and some of them were very easy for our respondents as well. So for example, mostly all of them knew that light travels faster than sound, that the Earth revolves around the sun, not the other way around, and so on. But some questions proved to be not so easy for them, and those were mostly regarding the new technologies. And for example, they were reluctant to dismiss the fact that regular tomato has DNA while genetically modified does not. Or they also were not sure that we share more than half of genes with mice and so on. So some easy, some not so easy for our respondents. So when we looked into the educational differences, those were the questions that we observed the highest educational differences. So there's something in the formal education that actually informs the participants about this. The age differences were also the highest on these type of questions. So the younger ones were more exposed to these types of knowledge. When you put this into context of other countries where on representative samples similar questions were posed, you can see that Serbia is either in the middle of the list or even on top of the list for some questions. So yeah, good news, so to speak. Scientific knowledge, at least the basic one, is there, and it's on par with the other countries where it's been tested with the similar tests. More good news, people are willing to learn. So when we ask them whether they want to learn more about scientific findings, at least declaratively, they say yes. And when we ask them to assess their own scientific knowledge, one good news ran away. So when we ask them to assess their scientific knowledge in comparison to average citizen, they tend to underestimate it, which is quite unusual for psychological finding. So we interpreted this as a room for education. People are kind of signaling to us that they want to learn more. More good news, trust in science. When we ask them whether they trust in science, they say they do, overwhelmingly, and that's because scientific method is a reliable way to learn about the world. Those who do not trust it say that it's basically because its findings keep on changing, which also signals to us that we should investigate how to communicate scientific uncertainty to the public, and we are now looking into it. We're conducting experiments on that as we speak. We also try to disentangle that trust-distrust issue about the science, and we ask them whether people trust scientists more than they should. People disagreed. We ask them whether our society places too much emphasis on science. Hard no, no. We put too much faith in science, still disagree. Here they were a bit more reluctant, but still disagree. A lot of scientific theories are dead wrong. So far, so good. There is this little caveat. We ask them about science in general in most of those questions, but when we ask them about specific topics, the picture is a bit more complicated. So science in general, very positive. Climate science, very positive. This is something that's not politicized nor contested so far in Serbian public opinion. But then biotechnology, GMO, and nuclear energy in particular, not so highly evaluated. And this is more of the same. So there is solid evidence that the rise in the world's temperature is due to human activity. Majority agrees. But then genetically modified food does not threaten the natural order of things. No, they don't think so. And then more nuclear power plants should be built to provide electricity. They don't think so. So some topics are still controversial and people are reluctant to endorse it. But okay, in general, those were good news, right? So decent scientific knowledge, willingness to learn, trust in science, at least in general. But people are complex beings, and these beliefs coexist with some that we call, let's say, epistemically suspect. Those are without solid evidence, very resistant to change, to counter arguments, and so on. And people tend to believe in both. So we looked into conspiratorial thinking, for example, and this is very fresh, representative sample in June 2023, and you can see that people do not dismiss those claims or they even endorse it. For example, a cure for cancer has long been found, but pharmaceutical industry keeps it secret from the public, or doctors prescribe untested dangerous medicines in collaboration with the big pharma and so on. So people are not protected from those unfounded beliefs via formal education, via scientific knowledge, even though they trust in science, they do not dismiss these types of claims. We as psychologists found that puzzling, that coexistence between those contradictory beliefs, and we also focus on this very narrow issue of endorsing really logically contradictory beliefs. And the COVID pandemic was ideal to study that because there were like very really logically incompatible claims that were out there. For example, doctors are paid to supposedly treat COVID, but hospitals are actually empty. And at the same time, hospitals are so overcrowded, patients sleep in toilets. So 6% of the sample agreed with both, right? And then like corona is a bioweapon manufactured for the mass depopulation, so very dangerous. And at the same time, it's nothing but a common cold, and it's very exaggerated. So 17% endorsed both. And we observed it in a number of situations. So it was kind of a puzzle to us. And we labeled those, the type of belief system, high entropy belief system, which allows like a lot of flowers to bloom, right? Like there is no place like to revise a belief, but they kind of let it be true at the same time. And this is the relationship between those types of beliefs and a lot of trust in science, but also some kind of health behaviors, for example, vaccination readiness. And you see that that conspiratorial thinking really has detrimental effects on all the other, so to speak, positive beliefs. More bad news, people endorse very strange health-related myths. Some of them are really obscure, like red drinks improve hemoglobin levels, incorrect diet makes food rot in the body. So they're not equipped to say, no, that's really not true. They're kind of allowing it to be true, maybe. So that can be just scientifically interesting, but what we find is that it also leads to a lot of risky practices. For example, when it comes to health, it leads to vaccination hesitance. It leads to non-observing the COVID-related measure. It leads to non-evidence-based health practices. For example, some of them are religious, and you see that prevalence is really high. Like 2/3 of the sample drank holy water to heal themselves. They prayed for their health. They visited monasteries and so on. But also some very obscure practices like lead pouring, which is not some, I can talk about lead pouring later, but it's still like 14% of the sample that went to a person who poured lead to get rid of their anxiety and so on. So just a very, very culturally embedded superstitious ritual. And also like some new agey stuff and like a lot of different alternative therapies, the prevalence is quite high. So we see that those are really entangled and that those, so to speak, epistemically suspect beliefs have their consequences. What we did with those findings, we tried to publish as much as possible to communicate with the international scientific community, but also to local stakeholders. This is, for example, analysis of how Serbian media portrays traditional complementary alternative medicine. Hint very positively, very uncritically, with a lot of pseudoscientific jargon, appealing on tradition, appealing on naturalness. This is more about this paradox of endorsing contradictory beliefs. This is about relation between those irrational beliefs and COVID behavior. This is more of that. And this tracking, we also track those questionable health behaviors on day-to-day basis. And we try to put them in concrete policy recommendations. For example, how narrative interventions can be used to counter anti-vaccination conspiracy theories, misinformation, how to label herbal and supplement industry to basically challenge their image as harmless, non-profit, and so on. What if we call it big supply? Would that kind of change the people's image of it? And we also contributed to this WHO edited volume about managing infodemic in the 21st century. So that's basically what we do. I hope you're not overwhelmed. I try to put it very, very briefly. And we have a lot of results at the both lab and projects websites and the social media. I'm here also for the rest of the panel. Thank you.
Thank you very much, Iris, for all of these results. This is really a lot of work, really. I don't know where to start. Well, there was very interesting data here about how they don't understand how DNA works and how it, and specifically in this context. And we were also speaking today that this might be something that is very specific to our kind of holes in knowledge from elementary and high school education. We'll see where this comes from later, but let's start with like what we know so far. And I would like to include Amy here to tell us about how the mechanism of RNA actually functions.
So RNA actually, yes, does many different things naturally in our cells, and I think you can think of it in sort of a few categories. One, we know that it is involved in taking information from DNA, which is sort of the blueprint or the recipe book in our cells, in all our cells, and it makes a sequence that then gets put into a protein. It makes a protein, encodes a protein. So it takes the blueprint, but actually makes a molecule that says which protein to make. And that's really important because it's actually quite dynamic, that we know that some cells have to make different proteins than others. Some cells need to turn proteins on when you have a viral infection or a challenge. So RNA is really key and dynamic to letting our cells respond to different environments and function. But it can also then regulate exactly how much protein gets produced. So what I actually studied as a grad student is how RNA has evolved to do so many different important things in our cells, and we're still learning all the ways that it very precisely controls the gene expression in our cells. And so it's involved in making proteins, it's involved in turning them down at the right time, and it's also involved in exactly which kind of proteins get made. And one of the newer areas that we're studying that's relevant also to therapies is the fact that RNA is also involved in transmitting information between cells. So it's fundamentally not just involved in allowing cells to function and respond and grow and turn into different organs, but also communicating between cells. And all organisms have RNA. Some people think it existed before proteins or DNA. It really has evolved to be able to carry out many important things in our bodies. And then, so it's a natural molecule that does many things in all organisms, in all of life. And we're learning how to harness that for different applications.
Some of those applications actually are in the sense of protecting us from virus infections, right? So can you tell us a little bit more about this mechanism and how it functions?
So yeah, there's a few ways that you can think about RNA therapies in the context of virus infection, and I study in humans, in mammals. So one mechanism that RNAs are involved in is, as I said, turning things off. So they are involved in shutting proteins down at the right time. And you can actually use this to target viruses, the same kind of idea. You can use a strategy that uses an RNA to turn off a virus. Viruses are not dumb, so they also evolve to try to use the RNAs in the cell. And another strategy that my lab has been studying, because we notice that viruses can actually shut off certain RNAs inside of a cell when they infect that cell. And that's because RNAs are part of our natural defense mechanism against viruses. So one of the strategies that we've been interested in is whether you can supplement cells with these RNA molecules as a way of boosting the defense to them and treating that infection. So there's many applications of RNA, and we're still learning all of them, actually. But it is a molecule that's in all our cells, all of life, and certainly viruses have evolved to try to take advantage of those molecules as well. So those are the nature of the areas where we can try to target viruses based on RNA.
Thank you, Amy. But it's also not restricted to humans. Some of the people, probably most of the people here are maybe aware of that, but I'm sure the general public is not. And Stephen is actually working on some fields that are not connected to RNA research in humans. And can you tell us more about that?
So, yeah, one thing with switching off genes, it works, switching off RNA can work very different depending on the different organisms. So everything, as Amy said, has RNA, but for particularly things like viruses, they're targeted very differently in plants than they are compared to humans. So something that I focused on in the past is protecting crops from viruses initially, and that's by using a form of RNA called double-stranded RNA. Viruses themselves often have double-stranded RNA genomes, so instead of having one strand of RNA, they have two strands, sort of like DNA, so very, very similar. And by putting in a matching piece of double-stranded RNA to a virus, a plant can actually switch that off itself, and that's its own defense mechanisms. They're very different to humans. They don't have antibodies, and they don't have that kind of response. They have a very specific response. So we basically use that mechanism to put RNA on. You can just spray it on. You can apply it in a number of different ways. And if that RNA matches a plant virus, the plant can switch off that virus and basically be protected against it. And in one sense, it's very different to humans. It's not like a vaccine where you can put it on once, the plant remembers. It doesn't have a memory system. It needs to be constantly reapplied. But that sort of approach is also effective against insects. And there can be a major crop protection issue against some kinds of insects where it works quite effectively. And it's also effective against fungal pathogens. So a lot of the work I'm involved with at the moment is targeting various fungal pathogens of crops, because I think if you look at crop losses worldwide through to the different pathogens, fungal pathogens are probably the major ones. And we've had a lot of successes so far with certain pathogens called rust pathogens. And it's one of those ones where we can just put it on a plant, that rust pathogen is completely gone after a very, very short amount of time. And we're trying to translate that through to a really wide variety of pathogens that are causing crop losses worldwide. And a big advantage of it is that it can target that pathogen very, very specifically. So when we put it on, it will only target that, and it can avoid all beneficial species. So the obvious beneficial species is us, humans. Doesn't have any sort of sequence matches to humans. But we also add things like honeybees, monarch butterflies, because they can be susceptible, and it doesn't target those. But particularly when we have insect targets, there can be very closely related insects that are normal pesticides that's currently used would just kill everything. So they're broad spectrum. They'd kill the insect that you want to kill, but they would also target everything else. With this kind of approach, we can be very, very specific. So we can have our pest insect, but even closely related beneficial insects, provided the design is done correctly, can be avoided. So a lot of my work is to use computational approaches to design these molecules. So that's exactly what happens, that there are not unintended impacts to non-target species, and it's a very environmentally friendly approach to crop protection.
Thank you. That gives us another side to the story. And there was also a recent event that gave us more light on the RNA story from the perspective of the Nobel Prize, actually. And this was actually not something that came completely as a surprise, since RNA technology and mRNA technology in particular has been developing for years, but has not been in the focus nor in the spotlight until the pandemic. But what does the story of the Nobel Prize tell us? Maya, can you tell us a little bit more on your perspective on this?
Yes, of course. But before that, I would like to maybe employ something that we learned today and try to explain for more general public what is actually RNA. Can you imagine your cells being buildings? And your organism is a city made of buildings. So the blueprint, as Amy said, is the DNA. So DNA is passed from generation to generation. However, who builds the building? So the RNA are the workers who build the building, and the bricks are proteins. So, but RNA is not just the workers who build, who help the build, but they're actually also guardians, ones at the door, defenders, all the structures that you could have in one corporate building, for example. All employees in the two buildings are actually RNA. So now we know that, but can you imagine that I don't know how many years ago we knew about the DNA, we knew about the proteins, what we didn't know how that information is transferred from the DNA to the proteins. And so the RNA was, I don't know, discovered in 60s or something like that. So until at that time, approximately, a little bit before, a little girl was born in Hungary in a very, very poor family. And she didn't have even electricity or running water, but she was curious. So she went to school, and after that studying biology and biochemistry, and it was in socialistic, communistic, actually, Hungary, where she did not have many opportunities for research. And finally, in 1985, being married and having a child of 2 years, her lab lost funding. So she was left out to work and didn't have any other option except to go abroad, which was terrible for her, losing her job and having a small child. But the sequence of terrible events that happened to her actually was beneficial to all of us today. So she went to USA to work in Pennsylvania State University when she worked as a regular scientist, and they were obliged to obtain projects. So she was working on RNA as this one specific type of molecule, those workers in the building. But actually, she was never able to be good enough in writing projects. She was good enough in science, good enough in envisioning or having vision, but not just in writing and obtaining projects. So finally, she was laid off. She lost her job again, and despite of her results and the promise of contained in those results, she envisioned RNA as being able to replace the protein, as Amy said, to replace something that a diseased cell lacks. So when you have a disease, your cells does not produce some kind of proteins, and she envisioned adding RNA to those cells to replace or giving more of those proteins. However, although her results showed that promise, she did not come to the final conclusion because there were many, many obstacles. And people should know that scientific research takes years and years, sometimes decades. And after being laid off, she found her next position in industry, which is again was terrible for her, but fortunate for us, because first she worked in one company and then switched to biotech. And in biotech, she worked with her colleagues and co-laureate of Nobel Prize to find finally a way to give the foreign or artificially made, because we can make RNAs in a machine, artificially made RNAs to the organism, to the cell, without, actually to the organism, without illicit immune response. That was not possible before her work. So we knew about the RNA, we knew about the possibilities, the promise was great, but the technical hurdle was big. So we cannot give it without big immune response. So she, being a chemist as well, biochemist, so she found a way how to chemically transform a little bit, but in the same way, the normal natural RNA that is in our body is always transformed. So she found a way to transform it in the lab so it would look like more of natural RNA, so it would be possible to elicit all the beneficial, all the promises, all the beneficial effects. And many scientists come to that point that they find something and produce it as a product finally. However, very few scientists in history have a chance to save the world. So this finding of hers was the basis for the design of the RNA vaccines, which were most powerful vaccines during COVID and the most distributed ones. So I think that just this path from being a girl from a very poor family without electricity and running water to the saver of the world is the fantastic path that we always have to have in mind when we think about science, but also think about scientists, because scientists are the basis of science. So the scientists, their knowledge are most valuable thing in the science. So their vision, their imagination, and their perseverance, because she could just give up on any occasion where she actually lost her job, but she did not. And that was, I think, very rightfully awarded by the Nobel Prize. Although I highly think of Nobel Prize, I think that the fact that she saved the world is much more valuable both to her and for us.
Thank you, Maya. That was a wonderful story, and it just is a great example of how storytelling in science is important and how we need to carefully think of how we present scientific results. And Rob here has done a lot of studies on storytelling and how we actually can change the narrative of the scientific research and how we can present it in a better way. Can you tell us something about the research you did and what you have found so far?
Sure. How long can I talk for? Like about an hour? One of the things I think that is not intuitive to people is that there is a science of communications. And for decades, psychologists and other social scientists have created hypotheses about how we should communicate better. I think one of the really interesting things that's happened recently, this research in Serbia is a good example, is it's become much cheaper to do quantitative research that's representative of the country to try and then check how these hypotheses work in the real world. And we have tested this for RNA to try and understand what is effective at communicating science. And I will not try and say everything, but I'll give some examples. Firstly, the stories of scientists matter. People have contradictory views about scientists. Sometimes they feel that scientists are irresponsible or risky or doing some things that they worry about a bit. And this appears in popular culture in places like Jurassic Park and Stranger Things and, Thomas will say, Bond films. But other times, people recognize that scientists are fantastic problem solvers. And so when we're communicating, we recognize that all of us human beings have contradictory feelings the whole time about everything. And so what we need to do is remind people of why scientists do what they do. And for the vast majority of scientists, they're doing it to make the world a better place. And Kathleen Carrico is a great example of that, actually. Somebody who was obsessive about RNA for decades, when people kept on telling her that it was a waste of time, but she kept on going for it because she was convinced she could make that difference. And that's a really heroic story. And that's also a story, though, that reassures people that scientists are doing things for the right reasons. And the general public care about motivations a lot, and they care about credentials very little. Your PhD, your professorship don't really matter, but the fact you care does matter. So scientists' motivations are important. It's also important, I think, to explain how RNA works and how these products work in a way that people can understand. We often talk about scientists being problem solvers, but we also talk about RNA being the language of biology. In other words, RNA is a way that living things communicate within themselves, between the buildings, really. We've also discovered, I think, that the biggest barrier to any expert communicating, including scientists, is their expertise, because it's very hard to remember what it's like to not be an expert. And so to communicate in very simple language is an incredible skill. So there's a trick you can do, which is you can get formulas that tell you how difficult is your writing to understand, and what age would a child have to be to understand it. And a very good writing can be understood by a 12-year-old child. So if you aspire to that, that would be good. I think most academic journals sort of break these algorithms, and they basically say you have to be 50 years old. But our aspiration should always be to make things as simple as possible. And I think the underlying reason in the brain is our brains use energy to understand things, and our brains are a bit lazy. And basically, when something is familiar, or it uses metaphors that are familiar or examples that are easy to understand, the brain likes that. And the brain confuses easy to understand with being true. One very sad counterexample you will all be aware of is Donald Trump. Donald Trump is obviously a terrible person and a complete idiot. On the other hand, because he communicates in very simple language, he is a highly effective communicator. And I did a study some years ago when he first got elected. His average speech can be understood by a 10-year-old. It has a lot of repetition in it. So even if you're not really listening, you have lots of opportunities to hear it. And it's very, very easy to understand. He doesn't say a lot in a speech because there's a lot of repetition. Hillary Clinton's speeches, they weren't bad, actually, but they probably required a 16-year-old to understand them. And that meant there were a whole load of people who weren't paying much attention, who understood what he was saying and didn't understand what she was saying. And I think a general thing we can always do is simplify, simplify, simplify. And if you want to see in the corporate world, this is why the best advertising tends to have a very simple story about something.
Thank you. So simplifying is one thing, motivation is another thing. So as researchers, Amy and Stephen, what motivates you to work on what you do in your labs? Can you tell us a little bit more on what you do and what makes you continue doing that? Amy, can we start with you?
I think two things motivate me. One, and this is true of a lot of scientists, we are curious often, but I think good-natured often too, and wanting to solve problems. And so to me, what motivates me with RNA and this concept of RNA communication is it's really, when we try to treat disease and understand disease or treat infections, we need to understand what's happening. And one of the things that we missed for decades is indeed, I think, the fact that our RNA molecule, that the extent to which cells are talking to each other and the extent to which pathogens can also get in there and spread fake news to our cells. So I think what I've always liked with science is where I can help, there's a gap and we need to figure out how to address it. And you have to be a little creative and maybe not too worried about how long it's going to take, and figuring out how we can go forward and how we can answer an important question. And I think with this concept of cell-to-cell communication and the role of RNA, it's incredibly timely, and I think it's really important. I think the fact that RNA is naturally moving between different cells, and we haven't understood that language. We haven't understood what information it's transmitting, and that's very much, I'm sure, underpinning disease, including cancer. It's known that that is one way that tumors can send out messages and affect the environment of the tumor, and pathogens as well. So finding that gap in knowledge and finding strategies to address that, and then ultimately finding ways of designing better therapies for diseases that affect us. That's a big motivating factor, and it's fun.
Thank you, Amy.
Yeah, I would add to that. The fun's the most important part. No, personal motivation for me is I'm from a farming family in outback Australia, and we grew sheep mainly, and we have a big issue in Australia with sheep with blowfly. So we used to spray loads and loads of insecticides all over the sheep to get rid of the blowfly. And it's probably 30 years ago, those insecticides were banned as being too toxic to be used. And that made me think at the time, and for a long time in my career, there has to be a better way than currently existing approaches to protect, in this case, animals, but also crops as well. And to give another example, in Australia, everyone probably knows we have the Great Barrier Reef, sort of a huge reef just off the coast. And it's suffering major issues from both climate change and runoff from agricultural lands. And again, a big problem of that is things applied to crops that are not environmentally safe. So it's a real driving force to me. I would like to protect farmers and by extension consumers. And the third one is to protect the environment itself. And I think being a scientist, we actually have that opportunity to do that. And it's a major motivational factor. But I'd also come back, I really like science for the fact that it's interesting. You go to new places that, ideally no one's been before. And coming back to the first thing, it's fun.
Thank you very much. So we see here that both of you mentioned problem solving, actually finding gaps, finding new solutions for problems that we have, and finding better solutions than those that we already have. And we've seen in communication, these are also good frames to model effective ways of transferring, like what we do in our labs and why it is important. What other frames of, what are ways of framing information are helpful in this sense? What are we doing good in communication, in the scientific communication, and what are we doing bad?
I will start with the bad because it implies the good. The first bad thing that most experts do is they immediately, when they're asked about something, they talk about what they are doing. And what that misses most of the time is the problem they are solving. So it might be people who are getting sick with a viral illness. It could be flu or it could be something else. If you don't talk about the problem you're solving, nobody actually cares about your solution. And what it's very easy for experts to do is just to forget that for most people, they are not thinking about this at all. And so if you start talking about your brilliant new bit of RNA technology, it just doesn't seem relevant. And of course, the biggest failure of most communications in the modern world is not that they are counterproductive, it's that people don't listen to them at all. And if you want an example of this, I was saying to Stephen earlier, everyone in this room has probably seen 300 or 400 adverts today. but I doubt many of you can name more than one or two of them, just simply because we've all grown very good at filtering out irrelevant information. And the problem is our information about science is also irrelevant unless we are smart enough to grab people's attention. So the first thing is we have to talk about the problems we are solving. Secondly, as I said, we have to simplify it. And then thirdly, we have to give really granular, concrete proof of what we're doing. But that's not proof in the sense of an academic journal. It's proof in the sense of a story that people can remember. So the story of the discovery of penicillin is a scientist whose experiment got disrupted by an open window and some spores falling onto a Petri dish and disrupting his experiment, and therefore he discovered penicillin. Well, that's a great little story because it's something goes wrong. It's something interesting and fun in many ways. You can imagine he very nearly threw it out. And so it's what are the stories that can help people understand what we're talking about and what is specific enough and what is maybe sometimes a metaphor that is close to people's everyday lives, not just the way the scientists think about it. I think the final thing about all of this is all of this requires us to try and empathize with people who are different from ourselves. And that biggest problem of being an expert is that it's hard to empathize with people who aren't experts anymore. And the solution to that is evidence. It's evidence which could be quantitative polling, or it's evidence that can simply be grabbing the teenagers at your next family party and telling them what you do for a living and seeing if they understand you. And if they look completely confused, you might have pitched it too high. Keep on retrying. You can keep on testing things, even in that very informal environment. That will teach you a lot.
Yeah, if you can't explain it to a 5-year-old, then you don't understand it very well, right?
5-year-old sounds tough, but certainly a 12-year-old, I could too.
That's the line. Great. Well, we've obviously done a lot of wrong in communication in RNA in particular, since from the results of our recent study in Serbia, we've seen that not everyone is really keen on understanding how RNA works because there is still negative stigma attached to RNA technology from the COVID regime, and we still associate RNA technology with vaccines, although now we've learned that it's much wider than just the vaccines. But in order not to impose that this is the only way to use RNA and to develop it, but let's try to break up a little bit what was the problem with mRNA vaccine acceptance, and what was the issue of why people were not, Maya, you can tell us maybe something more about what was this gap of understanding and how we could have maybe communicated the story about mRNA vaccines better.
Well, that's a very complex answer to that question, and it's not a definitive answer. There are several aspects to that. So the first aspect is quality of education that is keep getting lower, basic education in elementary and high school, that is lower and lower all around the world. So that's one thing. The other thing is preparation of the public for the new things. So COVID break was abrupt, and public was faced with a new thing very abruptly without many preparations. So whenever something is new, people are scared. The first thing relates to not understanding how that works. Third thing was too small number of people explaining in lay terms how does that work. So the wrong people explaining how that works. Actually, in Serbia, many different professions try to explain both COVID and the solution, the COVID vaccines, to the public, but those people were of different professions, not really related to RNA field or vaccine development field or even immunology. There were mostly epidemiologists, which was okay for that part of the story, but not the part with the vaccines. And public and some of those people are actually spreading not really correct information. So the public was puzzled. So they received conflicted information from different sides. Of course, social media spreading all kinds of negative and not true information. So altogether, one average person did feel very confused, did not know whom to trust, because there were professors saying, yes, there are COVID, and professors saying, no, there is no COVID, doctors as well, politicians as well. So the average person who is confused doesn't know whom to turn to. So the identification of the relevant source of information is also a very big issue in public, because we didn't have any reliable source of information. And altogether mixed did not go very well in Serbia related to RNA vaccines. However, that was in the beginning. As time passed by, I think that, I think that the key factor for accepting was the wide acceptance all around the world and the numbers. So if you say that, I don't know, 11 millions of people were already vaccinating and almost none suffered any consequences, then people were slowly more convinced. However, I think that fact, those numbers were not stressed enough. There were room for even more pointing that out. So I think altogether it finally ended well. So finally people in Serbia did actually accepted RNA-based vaccines, not all of them, of course. But I think that now that all the people can see that those who received those vaccines are okay, and none of them died, and everything is okay. So I think that this technology holds a great future. So as Stephen here said, not only in the medical field, but also in the agricultural field, forestry, all the other kinds of living things. We can intervene in the future with RNA. So it is really important to prepare the public in advance and to actually demonstrate both our good results, our first products, and the beneficial things that come out of using them. Thank you.
Thank you. That was actually a great answer for two very difficult questions. And again, communication is key, but people still have their fears. People still tend to believe things that might not be true. These are psychological mechanisms still that are in place that we can't easily change. And what they believe is related to how they behave in the end.
Yes, but can you think about magnetism or electricity or radio waves or anything throughout the history of science? Public was always against it at the first, and now we all use it in our phones, and even when we sleep, we keep phones near us. And so that's the question of time. So we need a little bit of media effort, a lot of scientific effort, and a time. It will just settle in time, I think. think.
So it is still quite novel, although it has been developing quite a lot, but it is still something that we need to learn more about. And in the context of the results that we've heard in the beginning, what can we, well, can we make any solid hypothesis on how people would accept RNA technologies more, or if they ever will? Can you tell us from your own perspective?
I can try. So what we can expect is a lot of fear, a lot of moral panic, which we already observed, and a lot of stigma. The thing is, yes, that's basically the cycle of adopting all new technologies works like that. Like it starts with reluctance, with a lot of moral panic, and then eventually it gets mainstreamed and people new generations are just faced with it being used all over the place, not having to adopt it. So there is that, like time will probably heal the wounds that we have now. But I think what COVID has learned us that if something requires cooperation from the public, we need to engage with the public, prepare the public for it, and not to expect them to behave fully rationally just because we say it's good for them to do something. Yes, and then like what we also learned is that people are not very good at distinguishing epistemic authorities. So they do trust scientists, but they also trust non-experts when it comes to that, when they're not really expert in evaluating sources, in, I don't know, the digital literacy is still quite low all over the world. So we have to take this into account when we think about how the adoption of the thing will go. So I think that the main issue about that was that we kind of expected people to go along because it's been a major breakthrough. It's like, it's saving the world actually. But then we have like people just denying COVID exists, watching other people die from it, right? So we shouldn't ever underestimate the power of denial, and we shouldn't ever expect people just to adopt facts because we just lay out the facts to them. So I think those are lessons learned. Will we actually employ them? It's up to us. But I think preparing the public and communicating, expecting not only information, but also misinformation to bloom all over the place, and then try to inoculate people to the misinformation, that's something that we could think about doing. Yeah.
Yeah, let's hope we do that next time, or let's hope there won't be another pandemic.
About pandemic, about everything that requires public cooperation, right?
Right, yeah, definitely. Okay, I think we can round up this part of the discussion and involve the rest of the audience here at this point. I will go around reaching the microphone to anyone who has some questions, or yeah, sure. Let's give them some time to think. And while the others think, I can jump in and use this like moderator privilege. My question is, what was actually stopping the development of RNA technology, if anything was stopping it? Because I wasn't aware that it wasn't really developed up until the 60s, and then we have a rapid improvement from there. But what is stopping the development in sense of, okay, now we know how some things function, but what don't we know, and what's stopping us from knowing more at this point? That's a question for both of you, and also Maya.
As always with science, there's a lot of things we don't know. So we're still a long way from understanding functions of RNA in what we call non-model organisms. So we generally study things like that are very, very well known. We study the same things all the time, but take, for example, fungal species. There's tens of thousands of them, and they can behave very, very differently from each other. So that's still some time before we get a very good understanding of how RNA works right across all fungal species, and that will take time and effort. So that's a big part of that. I think, what's the other part of your question? I'm going to forget the first part.
Are there any particular obstacles that you ran into in your particular research?
So RNA for agriculture was a very interesting one. It was first shown to be effective, spraying RNA on plants for viruses in 2001. So that's 22 years ago. And for the first 10 years, even though the experiments worked beautifully, the research just didn't go anywhere. The original research group sort of switched to other focuses. No one else took it up. So the big problem of why it didn't go is just because people weren't interested. And then it wasn't really till about 2012, 2013, where people were starting to look for something other than GMOs for crop protection, because it was becoming clear, particularly in Europe, that GMOs just were not going to be accepted. And then this sort of earlier research got dug back up again, and then people started getting back into it. And by 2015, 2016, there was a massive increase on using RNAi in agriculture for research dollars and for citations in scientific journals. So probably the biggest thing there, again, was interest. Funding is always an issue. And then for this, it has to go to a point where there's regulatory support and public support. And both of those are required for any of these things to sort of be translated from the lab to the field in my particular case. And that's slowly changing. I think particularly in Australia where I'm from, the regulatory support is already there. The funding support is still difficult, as we all face funding. You know, it's a big problem with science generally. But probably the biggest thing that has really held things back is, again, it's just that interest in it and the requirement, is it actually needed? And we're seeing more and more in agriculture that we need safe and sustainable solutions. And because of that need now, then I think it's going to have a very good future.
I think one opportunity and one obstacle with RNA technologies and medicines in humans, the opportunity is that now because of the manufacturing required for the COVID vaccine, we can make, I think, RNA at a reasonable cost. I hope we can and think we can make that in a sustainable way. So that opens a real door to indeed being able to use different develop RNA-based medicines. One thing which I think we will figure out and we're learning about is how to get the RNA to the right place. So depending on the disease and the indication and the location of that disease, you need to get RNA to the correct cells, and you don't want it to go everywhere. And we're learning how to do that carefully. And again, that is where we can learn from the natural mechanism and the fact that cells naturally are moving specific RNAs to specific places. We just need to study that. In our example with the viral infection, many years ago, we started trying to deliver RNA to the epithelial cells in the lung where you get infected with respiratory viruses. And the challenge was that there's other cells in the lung, there's macrophage cells that will take the RNA. Well, that's not where it's supposed to go. And what's exciting is now we are finding new strategies based on natural mechanisms to get RNA to the right place. So it's the medicines in humans, it's a partnership between the RNA that we can manufacture, but also the delivery vehicle. And that's, I think, an exciting area that's being developed.
Thank you both. I feel that it is exciting because it will allow us for a more personalized approach to both medicine and crop protection and animal protection and even forest protection. Someone was actually telling me something about that today. Yes.
My question is, we're talking here about communicating science. We're talking about the subsection of RNA. If I ask the panelists what you think are at stake, is at stake here, and where would we like to go? When you're on a journey, it's always good to know what the destination is. First of all, what is the worst case if we do not well communicate about RNA well, and what is an example of a science or scientific concept that has been communicated well?
Thank you for your question. So this is a really interesting question. So I think that RNA, both therapies and other products, will be accepted eventually. If we don't communicate it well and in time, that period is just going to be prolonged. So similar to RNA vaccines, if there was not COVID, we would wait for those vaccines that could also be used for cancer, for example, or in the same technology for other purposes, not just vaccines. We would wait for that for maybe 10 or 15 more years. So the pressure that COVID elicited actually speed up things. So I think similarly, our communication can also speed up things, not as much as COVID. And that communication, and also related to your previous question, should go towards both regulatory bodies and general public. So we need acceptance from the general public, which would be reflected in the politics towards regulating this area, but we also need to communicate our findings and the significance of our findings directly to regulatory bodies. I think that is very important. Educating the new generations through the school, to the workshops for children is very important in the long run, but we cannot wait that much. That should be done, of course, but we also need something to be done in a shorter terms towards the regulators and those ones who will actually set the scene for this to work. So we need laws, we need regulations, we need procedures to go towards products. So in some countries they are already there, maybe not just for RNA, but general, I mean, of course, they're general for different kinds of drugs. But these biological drugs in many countries, especially RNA, don't have specific regulations that might be needed in order for us to actually put our products one day to the market and employ them. So when we have products, it might be late. Now it is the time for us to warn them that this is coming and they should be ready as well.
I like the question of what's a success story? What's a success story with other technologies? I don't have the answer, but I just, what went really well? Sorry, I'm repeating the question.
I can think of one. And it's back to the vaccine example. When vaccines were first developed, I think against polio, they went from a new technology that was a worldwide success, and then whoever was responsible for the communications back then did an incredible job, because it essentially solved the problem of that disease within the world. I know it's still around, and smallpox as well. That between the two of them, that's a massive, massive success. Now, it's only more recently, since we've had all the, the vaccine scares and all that sort of thing happen pre-COVID, that really got amplified during COVID. But definitely vaccines, to me, is one of the hugest success stories that's happened in the last, how long have they developed for? 70 years, I guess.
Similar maybe to the previous one, but I'll try also to give my comment. We heard a nice story about the Nobel Prize. So on one side we have this great technology, but I'm still wondering, we had that case in Serbia and then all over the world, that the adoption of the vaccination was pretty low. So is the communication the only way for the society to adopt this and maybe other technologies? And I'm sure that the regulatory side is probably not the only one, but what is the way besides communication and the regulatory to have the higher adoption and actually people using it and not having all probably anti-vaccine part of the society.
No,
I would say, I think communication always has to be a very large part of it, because even the strongest dictatorships struggle to force their populations to do very many things, actually. And so I think, like, at one level, in making sure people feel motivated and comfortable is a huge, huge part of it, of the job to do the whole time. I think the other thing can be done is also just make it easy for people. And I think it's extraordinary how little people think about making it easy to do good behaviours. So as an example, I worked on in the British programme to encourage people to stop smoking, where they discovered that they got about 5% more people gave up smoking when they simplified the app that helped you to give up smoking. Now, giving up smoking increases your life expectancy by some years. So spending an extra 10 seconds on an app should have nothing to do with giving up smoking, logically. But the reality is, actually just making things easy for people can be an incredibly powerful tool. So Amy, what was the example you wanted me to repeat? Was it to do with peer pressure?
The UK was a brilliant example of vaccine
uptake.
uptake.
So the UK, people, just a really interesting example of where communication is used really well. I mean, UK government has not been a particularly effective government in recent years. But in the COVID vaccine campaign, the really clever thing that was done was they employed very good communication scientists, some of which Thomas and I have worked with here. And one of the central parts of the campaign was to say to people like you are getting vaccinated. Now, it was helped by the fact that in Britain, all vaccination happened by age. So the first round was for people who are over 80 years old. And so if you were 82 years old and you were a bit skeptical, a few weeks into the campaign, all of your peer group had got vaccinated and were clearly absolutely fine. And that happened for each age group as it went down. But it was combined with a communications campaign that featured ordinary people being vaccinated, and it said things like 9 in 10 30-year-olds have now been vaccinated. And it didn't say this works, it didn't say it was safe, it just said everyone else like you is doing this. And actually that turned out to be a phenomenally effective bit of communication. Britain's vaccination rate has actually been extremely high, despite the fact that people have had relatively low levels of trust in the government compared to historic levels in Britain, despite the fact that Britain has had a lot of polarisation on other issues in recent years. So half of the population were instinctively a bit sceptical of what the government told them. All of those things were to the side. It was a very successful campaign. It wasn't even a particularly expensive campaign to do. So I think that's actually a really good example of where something quite simple can be used, but it wasn't focusing on the science, it was focusing on the sense that everyone was doing it.
Can I just add a little bit? So what we also discovered in our work is that people, especially scientists, are communicating the vaccination mood wrongly. For example, in Serbia, people would say like there is a lot of anti-vaxxers, which in fact were less than 10% of population, but saying like a lot of anti-vaxxers, it's growing stronger and stronger and so on. It's signaling to people that there's something to it because more and more people think like that and so on. So basically, I'm not advocating for telling the wrong things, but this was true. And it tended to be overblown, and the fears tended to be overblown, which kind of signaled to people that there's something to be feared of. So that's one very, very common mistake that people use, trying to fear the public to cooperation. And I don't think that actually works. And signaling descriptive norms, for example, people like you do this, it's way safer, and you do not engage in the scientific debates. You just say, people are doing it and it's safe. You can see that. So that's something that I think we should think about.
There is also one thing that I don't know how specific it is for Serbia, but in former Yugoslavia, scientists were highly regarded and highly valued, not just by general public, but from the government. So they were respected, they were mentioned in the public by the government. Government was proud of our scientists. They were highly paid, and they had a good means for their work, and they produced the results as a feedback, as a positive feedback for politicians to brag about. After dismantling of Yugoslavia, that situation declined, and now we are in a rather difficult position that is actually known to the public. Even if you don't know any scientists personally, you know that scientists are not valued anymore. So people know our salaries, our conditions, our buildings. So they don't see us as something of high value, and therefore they are reluctant to trust in what we are saying in our stories. And I think that is an issue that could only be solved at the state level. Even if we go out in the media, we would be perceived just as whining. That is something that we cannot communicate, except rarely like this. But we don't have anywhere to communicate, not even with our government. So unless that is resolved, unless we change the scientific politics, I think that in the future people will just, still think that and not trust us. And the new pandemics or some new technological development that should be employed is maybe around the corner. So there is no time to waste. And the system is big and really complicated. So I understand that it's really hard to change. But I think, again, that is the matter of the state policy.
Do we have any more questions at this point?
Can I just point out one thing in the last question I forgot to say? Thomas asked about what success and failure looks like, and I think it's very important to realize failure in a democracy can very easily just be a good technology not getting regulatory approval. So the worst case scenario is the technology never gets used for all the good things it could be. But I think your point is, what's probably more realistic in other places is it just takes a very, very long time to get used. And I think a lot of us will be involved in debates on things like climate change, where the deadline the world has set itself is 27 years away, with quite a lot of things to get done in that 27 years. And therefore, a month of delay is, about, what's that, about 0.4% of that time. A month of delay is a significant thing if you're tackling climate change. And I think delays are very important. And that's where there are lots of subtle things governments do that slow things down. And sometimes it can be as simple as the regulators don't have enough staff to consider things properly, and therefore they go slowly. And that can be just as damaging as other delays. On the upside, I do think it's also worth saying that nobody else here said, the benefits could be treatments against cancer. They could be treatments against genetic diseases. They can be vaccines for all sorts of diseases that we don't have vaccines for now. They can be environmental pest controls that don't kill bees and butterflies and are much better environmentally. There are very many benefits here. And we have to keep on actually remembering to say those to people, because for most people, those won't be obvious.
That's a great reminder.
I just come up with two things. I mean, I agree that our society is becoming much more complicated, and also how you communicate about science. But I think about 10 years ago, or in the last 10 years, the last decade, we are also faced to a problem of news and fake news. which is making extremely complicated. And making fake news is very easy with this little machine. So it's making it very complicated. You can blow up things, and sometimes people do not believe the real numbers, like the anti-vaxxers is 10% of the whole society, but we all know the one that shouts loud, boasts loud, is heard. So they get impact, although they should not, but people listen to that. And the other thing is that people also have changed, I think, in the last 10 years, again with this little machine, that everybody has an opinion. That was also true before, but everybody wants to be heard, his or her opinion. And I think this is something that we have to look to ourselves, that are we patient enough to wait until research has solutions? I mean, it doesn't come in 1 year. It doesn't come in 2 years sometimes. Look to the Nobel Prize, it takes 10 years, 20 years. Do we have patience enough? And do we have trust in our government or in our scientists or in industry that they work seriously? It says something about ourselves. And I think we should also look a little bit in the middle, where we also make mistakes in our judgment. And that brings me to the last thing. We have little perspective back in history. We think, oh, something happened last year. That is our perspective. And even then, we have a very bad memory. You should look back 100 years ago. Look to the discoveries of Pasteur. And then look to the vaccination. Look to the real problems that happened with Spanish fever in the First World War. And then look to the situation of the COVID. But the perspective that people have, and this is related to education, this is related to this little machine, again, we don't have this. And it's also not anymore in the, how would I say, how people live, how people talk about things. Everything is fast and everything is short term. Looking back in history, more than PhD students, when you look to their literature research, everything that is older than 5 years does not exist for them. So this is also something we have to look to our own society and to our own person. I know it's a little bit out of the context, but it's not only that scientists have to communicate, it's also the receiver has to look to him and herself. How do I accept or percept the message?
It takes both ends to communicate, of course. And since we do have a short memory, we could learn more from the examples from the polio vaccine and from other examples, not to reinvent new solutions all of the time. And also, just to add a little thought that we might look for situations or solutions that are like on a high level, how to change minds and how to explain things to a lot of people, while we can start from the first person next to us. And sometimes this personal approach can be even more effective than a high-level approach, because if every one of us reaches out just to one person with a lot of empathy, with a lot of understanding and a lot of patience, they can change the world one by one, right? Thank you for your time here. Yes, anything else?
Just something comes up in my mind. If I would ask in Belgium, just in, let's say I go to Antwerp and I walk in the shopping streets and I ask people, do you know Tesla? I think 99% or 99 people on 100 will say an electric car. Will say an electric car. If I would say Nikola Tesla, nobody will know. So it brings me back to here, to this country, that this was a country really with a lot of innovation in technology, but many people do not remember this. And it's not 100 years ago.
Thank you for this final comment, since we've come, well, basically to the last minute of our discussion. Thank you. Thank you all for coming. Thank you for joining us in this discussion. I thank all of the panelists for this wonderful talk we have today. I hope we left you with a little bit more enthusiasm, and if nothing else, this story will definitely continue. We have a long way to go. This is just one of the first steps, or at least in Serbia. Somewhere it's moving a lot more fast in the world, but we hope to continue these discussions and continue our research on these topics and to evolve, to find new ways to communicate science better. That's it for us. Thank you.